And there's going to be still a panel discussion at the end, so there'll be an opportunity to probe some of these questions further. So the last speaker of the day is Marty Blaser, who's going to look at effective early life exposures to antibiotics in development of life diseases.
It's just going to take me a second.
Yeah.
Nope. Oh, there we go. Easy. Good. Thank you so much. Thank you. And I'd like to thank the organizers for the opportunity to speak here. The last speaker of a long day filled with information and ideas. But a big advantage for me is that so many important things have been covered that I hope will provide a context for what I have to say. So the title of the talk is Effective Early Life Antibiotics on Development and Later Disease. But an alternative title, maybe simpler, is What Happens When Our Early Life Microbes Are Perturbed? So I'm mostly going to talk about obesity, but I'll talk about some other things as well. And this slide is from NHANES data looking at obesity trends among U.S. children and adolescents over about a 35-year period. And the main point is that the rates are going up even in the very youngest children. And this is evidence indicative of a wide variety of information that the seeds of obesity actually are beginning in early childhood. Now, today I'm going to propose a hypothesis that antibiotics have something to do with this, that widespread antibiotic use is fueling this. I'm going to present the theory. I'm going to provide some observations to support this, and then review a number of experiments in mice that will support that. And of course, this is Fleming discovering penicillin, and this discovery really fueled one of the great advances in 20th century medicine, which was the discovery and use of antibiotics. And because of their remarkable efficacy, doctors began using antibiotics more and more and more. So, for example, in 2010, according to CDC data, if we look at outpatient antibiotic use, there were 258 million courses of antibiotics, a rate of 833 courses per 1,000 population. That's 5 courses for every 6 people, and this has been going on every year. The highest rate is in children in the first 2 years of life. And because use has been relatively constant, we can do a back-of-the-envelope calculation about cumulative use and suggest that in the first 2 years of life, the average child in the U.S. has gotten almost 3 courses of antibiotics and about 10 courses by the time they're 10. Now, these are big numbers, but they're in fact consistent with many other studies that have been done. However we count it, antibiotic exposure due to prescriptions is enormous. Now, there's a lot of variation in antibiotic use, and there's variation at just about every level that we look at it. But here I'm going to show you at the regional level. The national rate is 833 per thousand, Northeast 830, Midwest 868, about the national average, West 638, much less, South 936. 50% difference between South and West. No 50% difference in serious bacterial infections. This reflects practice and culture. Now, it's interesting to me to compare these two CDC maps from 2010. On the left is the obesity map. Darker colors mean a higher proportion of people are obese. On the right is antibiotic use. And when you look at these maps, they look pretty similar. Now, these are observational data. They don't tell us anything about causation, whether A causes B, B causes A, or C may cause A or B. But they get our attention. Here are some maps from 2011. CDC changed the colors, but the relationships are still the same on obesity. And here is antibiotic use in children under 2, which is the group that I'm most interested in. And again, we see a similar relationship. These are observational data. Now, I'd like to discuss a theory that could possibly help us explain why these two are related. And this theory has to do with human evolution. So on the left is the evolutionary relationships of wild hominids based on their host DNA. And there's a certain topology to this evolution, a certain branching pattern. And on the right is the phylogeny based on consideration of the fecal microbiota. And if you look at these two topologies, you will see that they are congruent. And this congruence is strong evidence for vertical transmission, and it is also consistent with the idea of coevolution of host and microbes. And this is from work by Howard Ochman and colleagues. Now, another important study was done recently by Yatsenenko and colleagues in Jeff Gordon's lab. Maria Gloria participated, and Rob Knight, a study of microbiota development in 3 populations in the world: people in the US, Amerindians in South America, Malawians in Africa. And on the x-axis is age. On the y-axis is the unifrac distance between children and adults. This is a measure of dissimilarity. Each circle represents the composite of the gut microbiome in one individual. And what the data clearly show is that when babies are born, their microbiome is very unadult-like. And then gradually and progressively, it becomes more and more adult-like until it is. And what's important here is that this transition is at the age of 3. The first 3 years of life are the most important time for the development of the microbiome. It is the most dynamic. It's the period in which the baby is developing. They're developing their metabolism, immunity, and cognition. It's also the period of time when they are having their highest antibiotic exposure. These ideas are consistent with a hypothesis that Denise Kirshner, a mathematician, and I had a number of years ago, that is illustrated here by this schematic of the interaction between a co-evolved colonizing microbe and its host. And the idea is that coevolution selects for microbes that know how to talk to the host and receive signals back, and that this is a form of dynamic equilibrium. Denise showed that this was robust and resilient. And what I want to discuss is what happens when these ancient relationships are perturbed, or in fact, if they're lost. So as Gloria and Margaret and others have told us, there is this transfer of microbes from mothers to children. There are many effective mechanisms. Mother Nature is very clever, but the mother of today is different than the ancient mother. Mothers of today are in an environment with antisepsis, antibiotics, diets with antibacterials. Babies aren't the same. As we know, about a third of the babies born in the United States are born by C-section. They're missing that ride through the birth canal. They're extensively bathed with antibacterial substances. They're getting formula, which doesn't have the selectors for beneficial organisms that breast milk has, and of course, lots of exposure to antibiotics. So based on these considerations, over the years, I've developed an idea that I've called the disappearing microbiota hypothesis that has 4 tenets. Beginning in the late 19th century, changing human ecology has dramatically altered the transmission and maintenance of our indigenous microbiota. These changes have affected its composition. This altered composition affects human physiology and thus disease risk. And loss of ancestral bacteria, usually acquired early in life, is especially important because it affects a developmentally critical stage. Several years ago, Stan Falco and I enlarged this hypothesis, which is shown here, the effect of maternal status on the resident microbiota of the next generation, with the idea that ancient moms had an ancient microbiota. If they lost organisms before they gave birth to the next generation, they could get them back horizontally, but if they couldn't, the next generation might be born at a deficit, and so on and so forth. In this model, the loss of microbiota is cumulative across generation. This is a very important point if it is true. Unfortunately, there is increasing evidence that it is true. Here we're looking at an indicator organism, Helicobacter pylori, an organism that has been the dominant organism of the human stomach for at least 200,000 years, probably longer. 3 generations of families in Japan, we can see the same kind of stepping down. If we look at the work that Gloria showed us earlier today, if we compare people in the US in terms of total diversity, and here we look at the uncontacted Amerindians and then these village people in between, we could almost imagine a similar kind of step. Based on the diversity shown here, it is consistent with the idea that we have already lost 50% of our diversity. So what I'm concerned about is the perhaps perfect storm of a cumulative loss of diversity and now a series of early life exposures to antibiotics. Now, our work in mice is based on the observation on the farm that giving low doses of antibiotics, subtherapeutic antibiotic levels, which we call STAT, promotes the growth of livestock. 70 years ago, farmers learned that if they did this, their farm animals would get bigger. That's why it's used so widely. It works from chickens to cows, and beyond that, it even works in insects. And so that's a wide swath of evolution, much greater than the differences in human genotypes. It works with just about any antibacterial that has been used, regardless of its chemical structure, class, target, or spectrum. Importantly, the investigators found that the earlier in life it is started, the more profound the effect on growth rate and feed efficiency, the ability to convert food calories into body mass. So we began a series of studies in which we gave mice antibiotics or not. We examined various properties, looked at the microbiome, sought relationships. I'm going to show you parts of 9 experiments that deal with aspects of this question. The first experiment was done by Il-Sung Cho when he was a fellow in my lab. He's now on the faculty of gastroenterology at NYU. Il-Sung gave 4 different antibiotic regimens, all in the midpoint of the FDA-approved regimens for growth promotion. And here we're looking at percent body fat, and we see that all of them, here's the control, all of them have enhanced body fat. And this is consistent with the idea here in this murine model that antibiotic exposure changed body composition. This work was followed up by Laurie Cox when she was a graduate student in the lab. She did an experiment that we called FAT-STAT, asking what are the combined effects of a high-fat diet and STAT on body composition. She gave mice low-dose penicillin or not. All the mice were on normal chow, and then at week 17, half the mice were put on a high-fat diet. We studied male and female mice. Here we're looking at total mass, fat mass, and lean mass. If we start with the male mice, this black line is the control group. No antibiotics, normal chow. On antibiotics, they're bigger, just like on the farm. On a high-fat diet, they're bigger still. High-fat plus antibiotics, big est. Lean mass increased on antibiotics, just like on the farm. Fat mass, big increase on the high-fat diet, even more with high-fat plus antibiotics. Female mice, very similar. I'd like to focus on fat mass. The average female mouse on the high-fat diet gained 5 grams of body fat. High-fat diet plus antibiotics, 10 grams of body fat. They doubled the amount of body fat. The antibiotic potentiated the effect of the high-fat diet. We see downstream effects on the liver as well. Now, in those experiments, the antibiotic was started just before birth. So Laurie did a next experiment where we compared starting antibiotics at this time just before birth or at weaning 4 weeks later, and we asked what would the phenotypes be, and our control group received no antibiotics. And here's one of the results. When we're looking at body composition at 20 weeks, we see the biggest effect is at birth. We see that consistently. And starting 4 weeks later, we lose most, possibly all of the effect. So it's clear that the timing of the exposure is very important, corroborating what was found on the farm. Now, in the next experiment, we asked, are the changes durable if antibiotic exposure was limited? Up to this point, the mice had received antibiotics for their life. So we compared no antibiotics, lifelong antibiotics, and only 8 weeks of antibiotics or only 4 weeks of antibiotics. Here we're looking at morphometry again, total mass, lean mass, and fat mass. The black line is the control group. What we found is that in all 3 groups of mice receiving antibiotics, there was an increase in total, lean, and fat mass. So 4 weeks early in life was sufficient. We were very interested in the interaction between what the antibiotics were doing to the microbiome, which I'll show you in a moment, and what was happening in the intestine. And we found a number of abnormalities, which I'll show you, but one of the most consistent abnormalities across multiple experiments were effects on intestinal Th17, T helper cell populations. So here we're showing work by Jackie Leung and Peng Lok's lab, small intestine, large intestine, flow cytometry, comparing control and stat mice. However we measured it, IL-17 is down in these mice. Now, what about the microbiome? Here we're looking at fecal community structure in mice at 3 weeks of age. And at 3 weeks, there are only 2 groups of mice. Control, not on antibiotics, or all 3 of the antibiotics groups are still receiving antibiotics at 3 weeks. Here in this principal coordinates analysis, the black dots are control, the orange dots are stat. We see that the distribution of positions is similar, but they're not identical. In fact, they are significantly different. That's not surprising. One group is receiving antibiotics, the other group is not. Now we're going to go to 8 weeks. Now at 8 weeks, we have control mice, no antibiotics, mice continuing on antibiotics, and we have one group of mice that got 4 weeks of antibiotics, and then they stopped. So when we look at the PCOA, we see the controls in black, stat in orange. They're more separated than before. Not surprising, they're continuing to get antibiotics. And the group that was on antibiotics but stopped, their microbiota have reverted to normal. So the effect on the microbiota is transient, but the effect on the phenotype is permanent. If we look at 10 weeks, we have a 4th group. Now we have the group that has gotten antibiotics for 8 weeks, and now they're off for 2 weeks. And we can see, again, these normal group, and these guys are normalizing. They're not quite normalized. By 28 weeks, everybody's back to normal except the group that has remained on antibiotics the whole time. So if we look at particular taxa, it's really most useful to look at colors. Here are the control mice. Here's where the high-fat diet begins, this black line. There's a bloom of this green organism. It's completely gone in the mice that are receiving stat for their whole life. With 4 weeks, we're seeing a delay, but still the same phenomena, 8 weeks, a longer delay. And what we found is that this is an organism we call Allobaculum, well, that is identified as Allobaculum, but it's probably some 2 new genera. And when we do a left-C analysis looking at taxa that are enriched in the first 4 weeks of life in multiple specimens before and during 4 weeks of life, we see certain taxa that are overrepresented in stat or overrepresented in control. When we look across experiments, we find several different taxa seen on multiple occasions that are down. We don't see any particular organism that is up across experiments, that is significantly up, suggesting that the major insult may be loss of organisms rather than gain of organisms. So at this point, we show this association of antibiotic treatment and phenotype and change in the microbiome. So now we do the transfer experiment where we ask, is the growth phenotype transferable by microbiota alone? The donors are mice that have received antibiotics or not. We take mice in the middle of the group so that there's no bias. They're sacrificed. Their fecal microbes are harvested in an anaerobic broth. These suspensions are given by oral gavage to germ-free mice, and now these mice are conventionalized and observed for the next 5 weeks. The study design involves donor mice that received antibiotics or not, with the harvesting of their microbiota, giving them to the germ-free mice that do not themselves receive any antibiotics. Here's the result on morphotype. Total mass, lean mass, fat mass, black line is the control. We found that the microbiota from the donor mice that had received antibiotics conferred an increase in total mass, no change in lean mass, and an increase in fat mass. These data are consistent with the hypothesis that the altered microbiota are sufficient for the adiposity phenotype. We also looked at immunologic properties, and here we're looking at expression of genes involved in ileal defense in the microbiota donor mice, and I'll show you in a moment the recipient mice. So we looked at genes involved in Th17 or downstream of Th17. The black are the control, the orange are the stat. We see in all cases that stat is down, just as we expected. These are the donor mice. What happens to the recipient mice? We see the same pattern as well. This study provides evidence that the microbiota are sufficient to confer the immunologic phenotype as well. Now, that's the kind of dosing that's done on the farm, but human children get an acute high-dose exposure. They get pulses of antibiotics to treat their ear infections and their throat infections. So we developed a second model, which we call PAT, for pulsed antibiotic therapy. And Yael Noble, a student in the lab, had the hypothesis that a series of short therapeutic dose pulses of antibiotics administered early in life will sufficiently change the gut microbiome to alter body composition. She gave 3 courses of antibiotics: amoxicillin, the most widely used antibiotic in childhood; tylosin, a macrolide. This is the mouse macrolide. This would be the Z-Pak for mice, or a mixture of these two. And taking multiple specimens, later they're put on a high-fat diet. I want to just focus on what happens to the microbiome. Here we're looking at richness and evenness. And we find that there's a big decrease, especially in the macrolide group. In fact, they never recover, at least out more than 100 days after the last antibiotic exposure. So based on those studies, Victoria Ruiz, a postdoc in the lab, asked, what's the effect of a single antibiotic pulse on gut physiology? So in addition to the 3-pulse group of tylosin, she also had a group that had a single pulse, and her control group. So here in this principal coordinate analysis, we're looking at the distribution of the microbiota at 4 time points. The first time point is day 22 after 1 pulse of antibiotics. Here's the control group in black. Here are the mice that received antibiotics. Both the 1 and the 3 pulse group have only gotten 1 pulse at this time. You can see that they are distinct. Now at day 30, it's after 2 pulses. And the group that has gotten 2 pulses has pulled away from the group at 1, but these guys have not come back. Now at day 39, we're after 3 pulses. They're further away. And and now 2 weeks later, these have come back to the position of the 1 pulse, but none of them have returned to normal. This is more than 40 days after the last antibiotic exposure in the 1 pulse group. Immunologically, we assess what's going on in the intestine using a hybridization technique, the nanostring encounter, where we're looking at 560 immune and inflammatory genes in the ileum, and we find 142 are differentially expressed. The black are the controls, and the colored are the 1 or 3 pat. They are all clustering together, significantly different than the control. We find genes involved in T cell differentiation, B cell maturation and maintenance, all down in pat. When we do flow cytometry and look at Th17, here's the control, down in PAT1, down further in PAT3, but PAT1 is sufficient for an immunologic effect. There's a decrease in the polymeric immunoglobulin receptor on epithelial cells in the ileum, so we looked at fecal IgA on day 27 after 1 pulse, down. On day 40 after 1 pulse, down. After 3 pulses, down even more. So we ask, is a microbiota necessary for PAT effects, or could this just be an antibiotic effect, a direct effect of the macrolide? So in this experiment, we gave germ-free mice PAT, the macrolide, or not, and as a control, we did the same in specific pathogen-free or conventional mice, control or PAT, and we sacrificed them 40 days later. So when we look at the specific pathogen-free mice, again, we see the decrease in Th17, and nothing is happening in the germ-free mice. They have no Th17 function. When we look at the nanostring again, in black are the conventional mice. These are conventional mice not on antibiotics, and by unsupervised clustering, they cluster by themselves. Here are the germ-free mice, whether they received PAT or not, they're virtually identical. And the mice that received antibiotics cluster with germ-free, not with the other conventional. So this study provides evidence that the microbiota is required for the effect. It's not an antibiotic effect. Finally, we ask the question, is the microbiota sufficient for the change? So for this experiment, we give mice antibiotics between day 5 and 10 of life, and then on day 12, we sacrifice control or antibiotic-receiving mice. We take their fecal contents and we put them into germ-free recipients, now conventionalizing them. We follow them over time. And here we're looking at IgA. This is work that Isaac Engstrand did when he was a visiting student in the lab. So here's fecal IgA. In gray is the control group. IgA starts out low after they're conventionalized, but then it goes up and reaches a plateau. But we can see that there's a big decrease in the IgA production. So we have evidence that there's an immunologic phenotype conferred by the altered microbiota. So for this phenotype, we have evidence that the altered microbiota is sufficient. So how do we put all this together? Our hypothesis is that antibiotics have an impact on long-term physiology through microbiota changes. The idea is that in the developing infant, there are still developmental decisions to be made by stem-like populations, and the gut microbiota is talking to them. And the basis of these conversations determine early life development of adipose, muscle, and bone. We've seen all these phenotypes as well as immune. This is health, and our idea is that antibiotic treatment changes the conversation, changes the developmental decisions, changes the long-term physiology. Lots of people did this work. I've mentioned many of them as I've gone along, but I want to point out Il-Sung Cho, development of the STAT model, Laurie Cox, FATSTAT, and others, Shingo Yamanishi, gene expression, Yale Noble, Pat, Alex Alexienko, informatics, Victoria Ruiz, the new immunologic studies. Lots of other people helping in different areas and funding from a variety of different sources. We've participated in 2 epidemiologic studies to ask, can we translate these changes in mice to observations in humans? And these are both based on the ALSPAC cohort in England, a cohort of about 10,000 healthy children born in the early 1990s. In the first study, we asked, is there a relationship between infant antibiotic exposures and early life body mass? We found that there was. Those who were exposed to antibiotics during the first 6 months of life had increased adiposity at 38 months and 7 years of life. This is one of 4 cohort studies showing the same phenomenon. that have now been published. We also looked at cesarean delivery, and we have found the same phenomena, C-section versus normal. And this is also one of 4 studies, and maybe we've got a 5th study from Margaret. So obesity isn't the only disease that's increasing. We have lots of other diseases going up in parallel. We've heard about asthma, type 1 diabetes, reflux. All of these diseases are currently epidemic and would be consistent with the idea that the altered early life microbiome has an effect. So I'm thinking that doctors of the future, when they're going to be examining babies as part of their well baby exam, in addition to examining the baby, they're going to examine the baby's diaper. And they're going to analyze whether this baby has the global microbes that all babies need or personal microbes that that baby needs and administer them and then figure out, is it working? This is somewhere in the future. And I want to thank my colleagues. And one last point, and that is I've written a book about this for the general public, and I unabashedly plug it. Thank you.
So, Marty, that was very, very, very nice and exciting. I kind of have a question a little bit about window of susceptibility. You showed in your mouse data that it was very much in the neonatal period that was very important. And, but many of the immune developments that occur neonatally in mice are actually occurring in the 3rd trimester in humans. So I'm wondering if you know if any of the studies, if any of the epi studies have actually tried to look at maternal antibiotic use, which often occurs, especially if there's a risk of premature delivery because of ruptured membranes or something like that.
Yeah.
And what the outcome is there.
I think I've got several answers to your excellent question. The first, of course, is that this is a model. It's not exact for many reasons. It is to test the idea that antibiotics change the microbiome, and that has developmental consequences. There are epidemiologic studies indicating that antibiotic use during pregnancy is associated with increased risk of a number of outcomes, including obesity. And as we were discussing at lunch, there's also evidence that antibiotic use before pregnancy is associated with risk of allergy. So I think it's actually very important that future studies look at all 3 periods: early life, pregnancy, and pre-pregnancy.
Any more clarifying questions, Ron? You're going to have to get to them. Let me see.
Ron Hines, US EPA, ORD. That same period of life, the first couple of years after birth, is when the vast majority of enzymes involved in xenobiotic metabolism also, you see a dramatic increase in expression. And yet we see a tremendous amount of inter-individual variability at actually when that occurs. And I was curious whether or not that might be worthwhile looking at, is to see whether or not there might be a relationship between the microbiome, antibiotic use, and controlling that phenomena.
Mm-hmm. Yeah, that's a very interesting point. I hadn't considered that before. I don't think that antibiotics are the only factor here. I think that they are an important factor, and we're going to learn just how important they are. But I think it's really about the nature of the ancestral microbiota and how we are perturbing it and maybe extinguishing it. And so there are 2 phenomena. One is just extinctions, where organisms are gone, and the other are what I showed in the mouse experiment, these transient perturbations where certain organisms are suppressed and then they come back. And certainly diet and other xenobiotics are going to play a role there, and that could help explain individual differences. Obesity isn't happening uniformly across the globe. It's happening in many places, but some places are happening more than others.
Great talk. I got a question about the crosstalk. Kunlu from University of Georgia. I got a question about the crosstalk between the gut microbiome and the host stem cells. Can you comment what's the potential molecular mechanism which actually responsible for the crosstalk? Or in other words, how does the gut microbiome crosstalk with the host cells.
So that's why I want to work with you. I thought you gave such a great talk, and it's on my to-do list. You know, there are many different potential mediators, and we found evidence for differences, for example, in short-chain fatty acids as one. We found evidence for increased energy harvest by an altered microbiota. Another interesting idea is that in the developing animal, there has to be a certain amount of energy set aside for the development of immunity. But if you don't have to set aside so much, then you can put more energy into other things like adipocytes. Now, that still doesn't tell us what the messenger is. In that case, the messenger is just less antigenic load and antigenic burden. That's an immunologic argument. But I think that it is likely that there are important metabolites that are involved in this, and we're interested in understanding in the gut lumen, in the portal circulation. I think that's probably the most important locus there, and then everything else is downstream.
At this point, we're going to continue the discussion during the panel session.